Okay, so welcome to this next video in which we are discussing shear stress and the endothelium derived relaxation factor. Okay, so far what we've seen is that when the shear stress on an endothelial cell goes up, i.e. the amount of particles within the blood knocking against an endothelial cell, when that goes up, what it causes is an activation of this TRPV4 channel, which is a mechanoreceptor, which when that channel opens allows calcium to move from the extracellular fluid into the cytoplasm. And this calcium then binds to apocalmodulin to create calcium-calmodulin complexes, which then activate the eNOS enzymes, which are bound to the inner leaflet of the phospholipid bilayer via their palmitoyl and myristoyl groups. Okay, those enzymes then create nitric oxide, which will diffuse through the lipid bilayer of this endothelial cell to the smooth muscle cell that lines the endothelium of the blood vessel and cause it to relax, which will cause vasodilatation of the blood vessel. And when you open the blood vessel, then the shear stress, the amount of particles that are hitting and bashing into the endothelium, and also the force with which those molecules are bashing into the endothelial cell molecules, that will go down basically, and that's protective against damage to these endothelial cells. Okay, so that's the main pathway. What I want to look at now is some facilitatory pathways, and also I want to look in a bit more detail at some experimental evidence for the involvement of this TRPV4 channel. Okay, so we'll start with this experimental evidence for the involvement of the TRPV4 channel. Well, basically, there are agonists for this TRPV4 channel, which will bind to this TRPV4 channel and cause it to open, and there are also things which will block the opening of this TRPV4 channel. So we could look at those and see whether we can activate the release of nitric oxide via activating these TRPV4 channels artificially with our artificial agonist, and whether we can block nitric oxide release in response to shear stress using antagonists of this TRPV4 channel. Okay, so let's do that then. So let's firstly look at the agonists for this TRPV4 channel. Okay, so an agonist for TRPV4 is something that's often abbreviated to 4-alpha-PDD. Okay, now what does this stand for? Okay, well, it stands for something known as 4-alpha, 4-bol, 12-13 didecanoate. Okay, so let me just move this out a bit. Didecanoate. Okay, now let me talk you through this molecule. In order to understand what 4-alpha, 4-bol, 12-13 didecanoate is, we firstly need to understand what the molecule that is called 4-bol is. Okay, so we'll start by looking at what 4-bol is. Now, 4-bol is not the simplest structure in the world. It's got 3 rings, but 4 rings in fact. We will get through it. Okay, so we'll start off with this 5-membered ring over here. We'll draw the skeletal structure because it's far easier to draw its skeletal structure than draw its molecular structure. So skeletal formulae, you only show non-carbon atoms, and carbon atoms you just abbreviate to corners basically. Okay, so you only show non-carbon atoms, and you certainly don't show hydrogens just bonded to the carbons. Okay, so here's this first 5-membered ring. Now the 5-membered ring has a double bond here. Okay, it also has a methyl group coming off here, and then it has a double bond down here with oxygen, so a carbonyl group down here. Now it's then bound to another ring, which I'll draw here, which is an unusual ring. It's a 7-membered ring. So how do I draw this? So that's 1, 2, 3, 4, 5, 6, 7. Okay, so that's an odd shape. So you've got 1, 2, 3, 4, 5, 6, 7 carbons in this ring. Okay, now off here, off this link, off this carbon here, you then have a hydroxyl group, so I'll put that in there. And then you also have a double bond here, and also a methylene group with then a hydroxyl group coming off here. Okay, so there's a single carbon with a hydroxyl group coming off it down here. Then, off this double bond here, you then have another ring, which is a 6-membered ring, so a more normal ring. So I'll draw this here. So this is our 6-membered ring here. Okay, and you have a hydroxyl group coming off here, like so. So then we need to discuss off this 6-membered ring here. Well, basically you have 2 hydroxyl groups, and these are going to become important. So here's a hydroxyl group here, and there's another hydroxyl group coming off here, and finally there's also a methyl group coming off this one. Right, so we've discussed 3 of these rings now. Now there is a really unusual ring structure. There is a 3-membered ring over here. So then we add this on, so that's a 3-membered ring there. And then off that carbon that's at this tip, you then have 2 methyl groups. So it's kind of a cool little structure over there. So this now is the structure of 4-bol. Right, now let me also tell you that the carbon here with this hydroxyl group off is called the 12th carbon, and this carbon here is called the 13th carbon. So what we're going to do basically is we're going to take 4-bol, and ignore this 4-alpha, I don't know what that means. We're going to take 4-bol, and we're going to add decanoate groups onto the 12th and the 13th, onto these hydroxyl groups of the 12th and the 13th carbon. I think 4-alpha actually probably stands for some sort of optical isomer of this I think it's probably telling you what optical isomer it is of this molecule. Okay, so let's now look at this decanoate bit over here. So decanoic acid then. So decanoic acid. Decanoic acid structure is you have a carboxylic acid group right at the end, and it's a 10-carbon carboxylic acid group. So we're going to need 8 methylene groups in the middle. So let's draw these here. So here's 1 methylene group. I can't be bothered to draw all 8 of them, so I will put brackets around them and then put an 8 down there to denote that you should draw them out 8 times. And then I'll put a methyl group on the end here. So overall now, I have 10 carbons in this molecule. 1, 2 on either end, and then the 8 that are in the middle here to make 10. So this is the structure of decanoic acid, and it's a saturated, long-chain carboxylic acid. Now, we want 4-bol 12, 13 didecanoate. What that means is we're going to take 2 decanoic acid molecules and we're going to asterify them onto these 12th and these 13th carbons. Okay, so let me draw you now the structure of 4-alpha, 4-bol 12, 13 didecanoate. So let's revise our structure of 4-bol then. So 5-membered ring here, okay, and then you have this double bond here with this methyl group coming off, this carbonyl group here, a hydroxyl group coming down here, and then this 7-membered ring here. Those are 3 so far. 4, 5, oh dear, and it's going to go horribly wrong. How am I going to show this? We'll have to have it going off like that. Never mind. Then we have a double bond here. That's a carbon there. That's supposed to be a corner. Okay, and off here you then have this methylene with a hydroxyl group down there. And you then have a hydroxyl group coming down here, and you have your 6-membered carbon ring here. Okay, and I'm sorry it's now slightly differently drawn. You then have a methyl group here, and now you have these hydroxyl groups, which we'll discuss in a moment. And then you have this 3-membered ring off here with these methyl groups coming down there. Now, of these hydroxyl groups, what you have now done is you've esterified these carboxylic acids, these decanoic acids. So you'll now have this carbon here double bonded to an oxygen, so that's that carbonyl group here. So basically what we've done is we've taken this portion of the molecule here, okay, and we are adding it on now. So here is the rest of the decanoic acid. So this is the methylene group. Okay. 8 times. And then we all have the methyl group off the end here. Okay, right. And then off this carbon here, we'll do the same things. We'll have a hydroxyl group that was originally on this 12th carbon, and we're now going to esterify it with a decanoic acid molecule. So here is the carboxylic acid group, here's the ester link now. And remember the hydroxyl group, the oxygen here that's in the ester link, that will come from the alcohol group rather than the carboxylic acid group. Okay, so here is the rest of our decanoic acid molecule. So our methylene group, which needs to repeat itself 8 times, and then finally the methyl group on the end. So this here is the structure of 4-alpha 4-bol 12-13 didecanoic acid. 12-13 didecanoic acid. Or decanoate, sorry. 4-alpha 4-bol 12-13 didecanoate. And that's often just abbreviated to 4-alpha PDD. Right, now what does this molecule do? Well, basically it binds to the TRPV4 channel and it causes it to open. Okay, so that's going to then allow calcium to go into the cytoplasm of the cell, bind to calmodulin, and then activate our eNOS enzyme. So we would predict that this drug would produce, would cause the production of nitric oxide by our endothelial cells. So what we do is we get some endothelial cells in a petri dish and we put this drug on and see if that happens. So usually for these sort of experiments what we use is a cell line known as HUVAC, okay, which stands for human, H, umbilical, U, vein, endothelial cells. Okay, now this is a very cheap cell line, which is why it's used for nearly all experiments involving endothelial cells. The reason being that if you want human umbilical vein endothelial cells, all you need to do is go down to a maternity ward and ask for some of the umbilical veins that have been removed from during pregnancy. Well, during labour. Okay, so these cells, we have a good supply of them, basically. So they're used for nearly all experiments that you do on endothelial cells. So what you would do is you take some of these HUVEC cells and you expose them to this 4-alpha 4-bol 12-13 didecanoate, and indeed what happens is they do start to produce nitric oxide. So that nicely confirms our theory then. Okay, now another drug is RUR, okay, which stands for rufidium red. Now, rufidium red does exactly the opposite. It stops these TRPV4 channels from opening, basically. It's an antagonist to the TRPV4 channels. It's going to stop them from opening, okay? So what we then do is we try and stimulate our endothelial cells by shear stress. Now you might ask, how will you do that? Well, basically, what we will do is we'll grow our endothelial cells on a petri dish, our HUVEC cells, our human umbilical vein endothelial cells. What you can then do is get just a little sort of hose pipe, if you like, and you can rinse water over them. So you can set up like a little flow system of water going over your endothelial cells here. Okay, and what that will mimic is it will mimic shear stress on these endothelial cells. So you would expect these endothelial cells to start producing nitric oxide because their TRPV4 channels would open, and indeed they do. Then what you do is you put rufidium red on these endothelial cells and see if that inhibits the production of nitric oxide, and indeed you find that it does. So this is all going very nicely so far. Both of our pharmacological agents have done exactly what we would have predicted them to. So that's some nice pharmacological evidence in favour of our model. Okay, and in the next video we'll look at other pathways involved in this production of nitric oxide in response to shear stress.